Okay, I am recording, and it's about almost 5 o'clock. We're waiting for people to show up today for this review. Oh, here comes somebody. That's Kyle. So now you're in, and he's here. So I'm going to admit you in. You guys can kind of see it up there. Our review session from 5 to 6 before your math class. Sorry. Yeah. No. I know your math class starts today, but just for this little bit of time, I'm okay, who else is here? Shalander, there she is. Okay. I'm gonna go to my view. I don't want Kyle anymore. I get Kyle off of here. No, I don't want to do that. There you are, see? So here we all are. You're muted, but that's okay. I'm going to share my screen. So I want Kyle to be you guys can kind of see the screen that I'm showing. Can you see the screen that I'm showing, Kyle? Okay. Let's see if there's anybody else. So Shalander left. Sorry. Oh well, just me and Kyle still. Yeah. Oh, there's somebody. Hello, who's ever come in? Welcome. Okay. Can you see that? Yeah. All right. So I'm going to go ahead and get started. Oh, I think I have this on record, so I think we're all good. And this first slide here comes from the exam 3 review that's in the Canvas support pictures. I think I talked about this particular diagram before, where we're trying to show how the blood interrelates to all of these structures here, so all of these parts, right, that are here. And so far, we've been able to talk about this part, right, where we identified the respiratory membrane between the respiratory system and the blood. And we're starting to talk about this part right here, where the nutrients are going to be put into the blood. When we get to the urinary system, we'll talk about a filtration membrane and how it's built. And then overall, when we talked about the capillary beds, we were in general talking about how blood gets distributed, nutrients and waste get distributed to the cells through capillary beds. So that's kind of a, the blood gets transported in this circuit carrying materials, carrying the um, gases, the respiratory gases, as well as the nutrients and waste products. Come on. So we're going to do a little review of the cardiovascular system. These are some important questions that you can start with. Some of these you kind of saw in the quizzes this week, right? So when we talk about the formed elements, right, what are we talking about? What are the things that are formed elements? Those are going to be the red blood cells, right? The white blood cells and the platelets. And then for each one, right, it exists in a particular number, right? In white blood cell populations, we talk about two numbers. We talk about the percentages of white blood cells, right? But for all of them, we talk about the numbers of these cells in a microliter of blood. So that's one characteristic about them that you have to know. So just see if anybody else is here. Before we go on. Nope. Oh. Oh. Sorry, guys. Hold on. There it goes. Okay. So diapedesis, what blood cell is related to diapedesis? Right, white blood cells. What's another name for white blood cells? Leukocytes, yeah. So these are the ones that move from the tissues from the blood into the tissues, and the movement action is called diapedesis. What about the plasma? Okay. So blood has two parts to it. The formed elements are one, right? The other part is the plasma, right? And we actually can describe, when we talk about that, the parts of the blood, we know that if we were looking in a cylinder of blood, that we'd have this big portion here, right? That we would call the red blood cells, right? If we put it in a tube and we centrifuged it down, then this would be the red blood cells. And then on top, the very top would be the yellowish fluid, that's the plasma. It's not showing up well here, but that's the fluid portion. And there's really this really thin layer in between. That's where most of the white blood cells and the platelets reside. And this is called the buffy coat. So when we're looking at a cylinder of blood, a vial of blood that's been spun down, we can see the major groupings associated with it. And so the plasma, all right, is one of those portions. It's this fluid portion, it's a solution, right? And into it get dissolved all the things that get transported. So this is the page that describes the formed elements, right? So the main thing here is to look at these descriptions, right, and talk about what they look like in response to the right stain. So that's color. Right? What colors the cytoplasm, what colors the cell, what colors the nucleus. Right? The other thing here is the shape of the nucleus. And the size, I'm not really going to have you memorize these pieces of this here, you know, this difference. It's really, do you have a general idea which ones are the smallest, which ones are the largest between those, okay? So which is the largest cell of these? Monocyte, yeah, this one's the largest of them all, right? And then the platelets are going to be the smallest, and then there's going to be a range of size in between, kind of like t-shirt sizes, right? Small, medium, large, extra large, like that. Then the next thing you have to know is the numbers in that microliter of blood. So that's this piece right here. So for red blood cells, there's 4 to 6 million cells in a microliter of blood. Right? For the basophils, it's like 50 to 100 cells in a microliter of blood, right? In that quantity. So that's a big difference between those two. Then the last thing over here is this function piece. And knowing which each one did. So if I said that there is a person and they appear to have an elevated eosinophil count, right? And let's see, yeah, let's say they have an elevated eosinophil count. What could you claim as being a possible issue? Parasitic infection, yeah. So be able to apply that, you know, if they have an elevated this and an elevated that, what could that mean based upon what you know about their function, so that you're applying that information. Okay. So this talks about the origin of the cells, right? And they have one common stem cell, so that's the blood stem cell here. Right. And then they can divide into the erythroid stem cell, the myeloid stem cell, or the lymphoid stem cells, and that will lead to individual cell types. So it's the myeloid cell that ends up making most of our white blood cells, right? So these are all of the white blood cells and also the platelets. So when somebody has a cancer that's myeloid cell-based, stem cell-based, then they have a cancer potentially to any of these cells here, okay? When they talk about lymphatic or lymphoid stem cell cancer, then that's mostly the lymphocytes, okay? Because the cell that usually the problem is the one that's dividing, right? The cancer cell that's dividing. So it's a way to maybe kind of understand it better or put together why it's important to know where they originate from, where types of that, and everything. Why it usually comes up in the kinds of lymphomas that they have, because those are the blood cell cancers. Okay? Of course it is. I'm going to get my cord. Or I'm just getting my computer plugged in. Hold on. All right. Shalander. Okay, Shalander, I hope you can hear me. I'm starting to talk about blood cells. Just hang in there and keep watching or keep listening. You should be fine. All right. Remember, I'm recording this if you go away again. All right. Another thing nice about this particular figure is that it kind of shows you what they do in relationship to each other as well. So we know gas transport is here, and which gas is being transported by this erythrocyte? Oxygen primarily, right? So blood clotting goes with the platelets, right? And then the basophils, the white blood cells, right? This is that army of defense that's going to play a role in the general defense. So mostly they end up being phagocytes. There are some that are more phagocytic than others, right? The neutrophil and the monocyte are more phagocytic than the others. The others generally will are weakly phagocytic. They do other things, like they release histamine or interleukins or some other things that help mediate inflammation. Okay. So any other questions about those cells? Okay. So plasma then. So when we talk about plasma, remember this is we've got all of these components that are in it, right? These are the various things that will be in it being carried, especially, after we've gone through that digestive tract. That's when we're going to see those nutrients go up. But really, there's a special set of proteins that you should know, right? And that's these different ones here, the albumin, the globulins, the fibrinogen, and that transferrin. So they are in the plasma, floating around in that, being carried within that plasma structure. And they each have a different job. So you should be aware what each of these jobs are for each of the different kinds of plasma protein. Okay, so I'm going to go stop the share. And let's see if I can get me to come on. I don't want Kyle anymore. So, not sure. Oh, there it goes. Okay, here I am. All right, so up here, I have some cards, and go ahead and take any one of them, any one of the stacks that you want. There's several of you here, and then see if you can just tape up on the board where the different pieces go, okay? So on this board, I've got the different white blood cells, red blood cells, platelets listed, and then looking at some cards, and they're gonna put them in under the right place with tape. No, don't worry about that part. This one, focus on that. See how many we can get done in about 5 minutes. So they're just putting up these cards, and we're going to go through them and see how well everybody did. good. Let's see what we got here. Okay. Hopefully you can still hear me. I'll talk loud so the computer will pick it up. So this is the erythrocyte column here. The oxygen transporting cell, that's true. Packed with hemoglobin, that's true. Biconjugate shape, that's true. And ABO blood markers. So all of that is true. So you got all of those correct. Good. Right? The blood cell markers are the ABO group and the Rh negative positive. But any blood markers are going to be found on the cell surface of the red blood cell. And over here, this says platelet. Right? And the platelets, they're not true cells, but they're fragments from the megakaryocyte. That's true. They don't have a nucleus. They don't have, they function in blood clotting. They don't have organelles. And that would also be true for the erythrocytes, right? They don't have any organelles. Okay. Come over here. All right, so eosinophil. So yep, that's the eosinophil. Right. Yep. So you got that one because it has the red cytoplasmic granules. So this is also true, composed of 1 to 4% of the white blood cells. These are the worm eaters, right? They play a role in parasitic infections and allergy reactions. So you got that. They're nucleus, and they have the red cytoplasmic granules. So that's all true. So that was good. You got all those. All right, let's keep going. And neutrophils. So this is true. That's the 40 to 70% of white blood cell population, 3,000 to 7,000. Then this is a neutrophil with the 6 lobes. This one has 1, 2, 3, 4 lobes to it. It increases in infection. It has 2 to 6 lobe nucleus, and these are the major phagocytes. So that's all true for that. Then over here, these. So basophil. So yes, less than 1% of leukocyte. That's true for that. Has 2 lobes to the nucleus, blue granules. Here's the cell, so that's true for it. And then these are the ones that produce histamine and heparin, right? That's the other thing that they produce to help mediate inflammation. Right, those 3 are in a group that we call granulocytes. Yeah. As one of the reasons they're grouped together is because they have the granules as well as this polymorphonuclear cell, right? Because they have these nuclei that have these money shapes to them. And then the lymphocytes are the monocyte. They're in the other group, which is the agranulocyte. So no granules in the cytoplasm. So we're mostly looking at the color of the cytoplasm and the shape of the nucleus here. So this picture is right for lymphocyte. That's what this one is. That's the second most numerous. So you got that right, 25 to 45%. So a large spherical nucleus with clear blue cytoplasm. That's true for that. And most important immune cell for the specific immunity. And specific immunity is where you build the T cells and the B cells, right? And they build a particular set of proteins, either through cell markers or antibodies that fight off infections specifically. And then the last one here, right, is the monocyte. So that's the characteristic monocyte shape with the nick. So that's good. And these are the phagocytic cells, and their numbers increase during chronic infection. So something like mononucleosis, TB. What's the one that elevates during short-lived infections? The one that's more common would be the neutrophil. Yeah, phagocytic during acute infection. And then 4 to 8% of white blood cells, that's true. And then the kidney shape of the gray cytoplasm. So all of that's true. So you got those. Good for you guys. All right, let's go back to the PowerPoint then. All right, so anything else on the heart? I mean on the blood? No? Okay. So for the heart, here are some questions to consider, right? Can you describe the valves of the heart, what their structure is, and how they work? So how they work is how do they prevent backflow? So for the atrioventricular valve, you have to include how the chordae tendineae and papillary muscles are a part of that structure to keep the valves closed so they don't evert. Okay. For the semilunar valves, right, that's the one where you'll have to just talk about how the gravity of blood fills them up. What are the heart layers? What's the function of each layer in the heart wall? Comparing the structure and function of the heart chambers. So this can be twofold, right? This question, when you think of it, it's comparing what the atria do versus the ventricles, right? But it's also the right chambers versus the left chambers, how they differ, go to different circulations. And then this structure part, right? Some have thicker walls than others. So why is that so? So there's really kind of three aspects to this structure function relationship. Just kind of the general, how do they work differently in circulation? How do they work versus pulmonary versus systemic circulation? And then how does the wall thickness make a difference and why? So this one should be straightforward for everybody, right? Going through the heart and knowing what part goes to what. So cardiac cycle is the rhythm of the heart initiated by the conduction system. And then when we talk about it, we talk about the two phases of the heart when it goes through the cycle, and those two phases are diastole and systole. So when we talk about that, we try to relate that to how the valves are working. Are they open? Are they closed? Right? How that relates to the sounds that get made, and how that relates to heart circulation. And then that ties right into this conduction system, because it's the conduction system that's going to initiate the periods of diastole and systole in the heart. Hello, sorry. So this is kind of an overview of all of the anatomy we talked about with regard to the heart. We looked at its location in the thoracic cavity, right, where it sits, terrible cavity, where it sits inside of the thoracic cavity, inside of the mediastinum there, surrounded by the pericardium and the parts of the pericardium, right? Then we talked about the parts of the heart wall, the chambers and their names, what vessels bring blood to the chambers, carry blood away, right? The names of the valves and what sounds they represent, the vessels that come off of the heart, the different pathway of blood through the heart, coronary circulation and conduction. So that's kind of a summary of all the anatomy in one place. So this is the heart wall piece. I'm going to just flip this to tablet for a minute. So the pericardium, right, is this part right here. And we have the serous portion. Oh, thank you very much. Oh, very good. Thank you. All right, so we have the pericardium itself, right, with the fibrous on the outside. So do you know what the role of the fibrous is? It surrounds the outside of the heart, right? It's going to help it to keep it from expanding and getting stretched out. It kind of maintains its size. It also helps to bind it in place inside of the mediastinum. It's attaching it to the top of the diaphragm there. So those are its two roles. Then we have the serous. We talked about the serous membrane being continuous, and then we have the cavity inside. And then that led to the different layers here. One of the things I talked about is how these cardiac muscle, right here in the myocardium, right, how the myocardium arranges in the atria versus the ventricle. And that's a structure-function relationship you should know. So how do they arrange in the atria? I have a picture. So in the atria, remember they arrange around this way, the myocardial cells arrange this way so that when the atria contract, they squeeze on the atria, a lot like our tunic layers do, right? When we have vasoconstriction or in the blood vessel, they're arranged in a circle, right, around within the wall. And that's going to push down and push that blood from the atria into the ventricle. And then here, there was this kind of twisting action in the ventricle wall because what happens is that the contraction is going to start at the apex and then work its way up each wall side here. And that's going to work to push the blood right into those arteries. All right, so basic heart anatomy, right? You have to know the different parts of the heart, starting with the veins and where they drain from, right? And as they carry blood into the different chambers of the heart, past which valves, where it goes to the different, into the pulmonary circuit, where it goes into the systemic circuit. So is there any piece of that? Okay. So here we can see those heart valves in relationship to each other, right? And we have the anterior here, posterior here. So this is the tricuspid valve. This is the bicuspid valve. So those are the atrioventricular, and then we see the semilunar valves in the pulmonary and the aorta here, right? So this tissue that's anchoring the valves is important, right? That was that fibrous skeleton, yeah. And the fibrous skeleton is going to anchor those valves. It's also going to be the attachment point for this myocardium, right? This is where it comes in and attaches. And it's also, for the conduction system, it's a non-conducting barrier between the atria and the ventricle, so that the stimulus will travel through the conduction system structures rather through the cardiac muscle itself. So here's what I mean, right? We have the parts of the conduction system, and here's that pacemaker, right? The SA node. And if we didn't have the fibrous skeleton, we really wouldn't need all of the rest of this apparatus down here, all the rest of this stuff, all right? Because if we didn't have a fibrous skeleton, then the wave of depolarization, the wave of stimulus would just travel from the atria to the ventricle, because all the cells would be connected to each other in some way, right? Through those intercalcated discs. Oh, Dr. Phil. But because we have that fibrous skeleton here, then the only way that the stimulus can pass into the ventricle is through these pieces of the conduction system. And that means then that the atria contract first, right? And then the ventricles. Because this has a little pause in it, right? It pauses and it gives enough time for these atria to contract, and then it passes the rest of that stimulus down into the ventricle, and then it contracts, right? It stimulates the muscle and then it contracts. And that's what we can read when we look at that EKG, right? The EKG is showing us the path of that electrical event and how it then affects the different parts of the heart. So we talked about how the different parts of this wave, right, represent different parts of the cardiac cycle, right? So that this part here is when the atria contract, this part is when the ventricles contract, and then this is when the ventricle relaxes. So I don't care that you know so much the individual names of these waves right now. You're going to get to learn all of that in physiology in detail. But just that this relates to those pieces of these pieces of the wave relate to when the heart is going through the cycle. Okay. Anything else on the heart? All right. So lots of questions for the blood vessels here that we can kind of talk about. So things that we talked about, and I've just put these into question form, right? What are the tunics that form the wall of the blood vessel? And describe each of these tunic layers and the role that it would play. Okay. So the tunic layers are going to be the tunica intima, tunica media, tunica externa. What's their tissue composition? And that basically tells you what they do, right? Because the tunica intima is mostly the endothelial lining, so smooth surface over which the blood can pass through the vessel, right? The tunica media is going to be the muscular layer, and in some vessels it plays a large role in changing the diameter. And then the tunica externa is always going to be the vessel, especially in arteries and veins, that is going to help attach it to other structures, right, inside of the organ or outside of the organ. So this one here, distinguishing between the vessel types, this is where you would distinguish between elastic arteries versus muscular. Versus arterioles, then capillaries, then venules, and then veins. Okay. So when we talked about this, we talked about where they were in the body, right? How much blood pressure they endured, whether they pulsed or not, and then how the tissue composition changed for each of the tunic layers. Hello. Thanks. These are all old students. So like the elastic artery has those elastic lamina in them, more so than the muscular. Muscular has more muscle tissue, right? Arterioles, they change their tunics from where they begin to where they end, right? So there's characteristics that distinguish them from each other anatomically, so you should know those. And that ends up relating to how much blood pressure they endure as well. So what are the mechanisms that return blood back to the heart? There's three, right? So which one? Venous valves, right? Muscular contraction, muscular pump, and the respiratory pump. Yeah, so those are the three that you have to be able to describe. Then the capillary bed. Let's see if I have a picture of that. All right, so here's our capillary bed. So we talked about the parts of the capillary bed, right? The terminal arteriole, the true capillaries, the post-capillary venule, and then this shunt down the center, right? Some of it's called the meta-arteriole, some of it's called the thoroughfare channel. So those are all the pieces, and we talked about the way this capillary bed would work, whether or not this sphincters were open or closed. So what would be the pathway of blood when they were open? What would be the pathway of blood through the bed when they were closed? So those are two ways to distinguish between those. All right, so this is where I talked about the relationship between blood velocity and capillary surface area. So that the whole capillary surface area is like one big open tube, right? That narrows down to a smaller tube, which we know is arteries. So the blood fat will travel faster in the arteries than in the capillaries. So that big surface area and all of those capillaries is kind of like one big tube that slows the blood down and allows for the exchange of material. So that's a structure-function relationship there. All right, and then this is what we're working on in the lab, right? The major vessels. So is there anything about that that you want me to review with you? See what I have here. About that? Stop the share. All right. Anything else? Is that what you expected? Do you need anything else from this session? Just trying to get you thinking about all of this. You've kind of done it as you got ready for that quiz. Was there anything about the material that you got asked about on the quiz that you wanted to verify or clarify? No? So hopefully what this did is give you an idea of where to put some focus into your look at the structures using those questions as a guide. Those questions, again, are online. You can go back through them and see, because I don't ask you those questions in full, but I do ask kind of pieces of them when I do your essay. And you'll see in the blood vessel question, some of that was in the worksheet that you did on blood vessels. Some of that was in those questions as well. So hopefully that will help you to be, it's more than just being able to name the stuff. You have to be able to understand how everything works, okay? All right, well, I'm gonna close it down then. And I'm gonna stop the recording.